So it's going online. I hope it's a bit slow. Before you continue, it's a new computer. So I'm pretty sure we are online now.
Yes, we are.
Yes. Okay, so good morning, United States, and good afternoon, Europe, and wherever else you are. My name is Carsten Schradi, and I'm happy to be the host of the last fine of the fine season 10 of spring 2025. And it's not only the last fine of the season, but it's also a new record for fine because it's the latest fine we ever had for the presenter, Geoff, who I will introduce in a few minutes. He's in Tasmania. It's 1 o'clock at night there. So it's a really highly appreciated from all of us that he's making this time here to present today. And but before I come to Geoff, I want to let me see. It's switching to this screen. Remind you of what we had last week. That was now you see yourself. Now you see last week's find. It was by Erin Sercusa on social aging. Great studies on these rhesus macaques on this island. Really fascinating. And there's no fine to present for next week, but here you see we have already the fine program for the fall season that will start the 2nd of September. It's nearly full with a lot of very interesting and fascinating talks. These talks include 3 people who in our Google survey we made earlier this year presented that they would like to give a fine presentation. We could not accommodate all of the people who indicated this yet. We still have a list of around 60 people we would like to invite, and we also, of course, consider the people who indicated they would like to invite, but not all of them can be at once. Another point I would like to make is that according to what we got from the Google survey, we included again more fine panels. There will be one fine panel 30th of September about writing for the public. This is about how to interact with journalists, how to write popular science articles or books. And in October, there will be a fine panel that's especially, but not only interesting for our young colleagues about the difference between a research project versus a research program. I will send this around next week. We have nearly presenters for every date, though we don't have all the titles yet, but you will already see it's going to be a very interesting season again with social fishes, multi-level societies in mammals, focal communication, and many other aspects. So stay tuned, and we will send around the program next week, and then the final program Eduardo will send around in August. But today's talk is by Geoff from the University of Tasmania in Australia. Like I said, it's very late for him, 1 o'clock in the morning, and we're very happy that he's going to talk today about social evolution in lizard. Geoff is a real Tasmanian. He did his undergraduate studies there, his master's and his PhD, always studying reptiles. And then for postdoc, he was collaborating between Oxford and Tasmania on interesting aspects of adaptation of reptiles to extreme climates. Interesting also for climate change, studying a species that had been introduced from the Mediterranean to England, so to too cold climate for it, and how they could adapt to this cold climate, for example, by extending the period that they were having the eggs inside them before laying them. And he liked Oxford so much, he stayed a bit longer for a Marie Curie fellowship to continue with these studies, and he's still collaborating with several groups in Europe. And he just returned from Europe on Friday, where he was in Sweden and in Italy. And we had hoped he could still present at the fine while he is in Europe, but now we're extending his jet lag by a few days by forcing him to present so early in the morning for Tasmanian time. And since he's back, when his Marie Curie fellowship finished, he got a lecturer at the University of Tasmania and still studying different aspects of the behavior of lizards, and also mainly on the social evolution in lizards. And that's what he's going to talk about today. And I'm very much looking forward to your presentation, Geoff.
All righty. Thanks, Carsten. I will try and manipulate this in a suitable way. Share my screen. I think should be this one. Does that work for everybody?
Yes, I can see it well.
I'm just gonna hide this panel. Alrighty, okay. Let's make sure this works. Oops. Okay, all right. Thanks so much, Carsten, for the invite, and thank you to everybody for coming along to today's talk. I'm going to speak today broadly about 2 decades of research that we have been undertaking, focusing on the evolutionary ecology of family living in lizards, as Carsten suggested. The focus of this talk is going to be very much on 2 different components. I guess first up, I'm going to pretty much describe what family living looks like in the lizards that we study, which is one group of Australian lizards, the Egernia group. And then I'm going to spend some time looking at reporting on some research that we've done to try and understand how family life has evolved in these lizards. So how these lizards have transitioned from what you might expect typical lizards to be, which is non-social living lizards, to simple forms of family life, and also how that has set the stage potentially from which more complex forms of social organisation, large communal family groups, but also more complex forms of social behaviour. So transitions to things like parental care have emerged once you kind of get these closely related individuals living together. Before I jump into the nuts and bolts of the talk, I've got a couple of acknowledgements. First, acknowledgement of country. So I acknowledge the Palawa people, the traditional custodians on the land on which I conducted the majority of the research that I'm going to talk about today. And I pay my respects to their elders past, present and emerging, and I recognise their enduring connection to land, sea and the community. The other important acknowledgement is obviously of all the people that have been involved in this research. This includes collaborators from all across the globe, as well as a number of postdocs, some of which I saw were in the audience, as well as PhD students and honours students that have obviously generated a lot of the data that I'm going to talk about. I'm going to acknowledge each individual as we kind of get to the particular bit of research that they've kind of undertaken. Some of this, unfortunately, hasn't made it into the talk. I had to cut a fair bit out this morning when it was quite over length, but I'm going to acknowledge them here anyway for their important contribution. I'm also going to highlight 3 people in particular that I'm not going to acknowledge necessarily as we go through the talk, but those are 3 people that have been involved in the research pretty much from the get-go, and that's Tobias Uller, Eric Wopstra, and Martin Whiting. So they kind of pervade this talk in its entirety. And also, obviously, acknowledge a number of funding bodies that have supported the research throughout its journey. Alrighty, so I probably don't need to give much of an introduction to this particular audience. Everyone's well versed in social evolution, but I will give a little bit of context. I guess focusing broadly on this idea that social behaviour and social organisation spans a massive continuum from organisms where individuals kind of rarely interact with one another, potentially only for the purpose of breeding, all the way up obviously to organisms where individuals live permanently in social groups. So there's lots of opportunity for social interactions occurring between individuals, and individuals live in these kind of stable, permanent social systems. Those social systems, those stable social systems can kind of emerge either through the kind of keeping together of closely related individuals in the context of fraternal societies, or then they can kind of emerge from the bringing together of unrelated individuals in the context of egalitarian societies. Now, irrespective of the route by which these stable social groups form, these stable social groups can form the foundation from which you get the emergence of a whole range of more complex forms of behaviour. So things like individuals becoming mutually dependent on one another, foregoing their own reproduction and potentially assisting others in the production of their offspring. And it's been argued that transitions to these complex forms of social organisation represent a major transition, a major evolutionary transition. And as a result, it's perhaps not surprising that evolutionary biologists have spent an awful lot of time trying to understand how these stable social groups form, and how they provide the foundation from which you get the emergence and evolution of these complex social behaviours. And to kind of address that challenge, researchers have used a whole range of model systems, social mammals, birds, some amphibians, fish, obviously the social insects. But I guess there's one group of organisms that have been pretty much underrepresented in these studies, and that is lizards. And this is largely due to the fact that lizards have somewhat been assumed to be relatively asocial. I guess that doesn't necessarily mean that they don't exhibit social behaviour or haven't been considered to exhibit social behaviour. Lizards are excellent model systems, or have been, for exploring a whole range of different social behaviours. So territoriality, mate choice, signalling, communication. It's just that they've been assumed not necessarily to live in these kind of stable social groups that we see occurring in these other model organisms, so in the birds, the fishes, the social mammals. But this is a perception, I guess, that is slowly starting to change and has changed over the last kind of 20 to 30 years as research across the globe has started to identify evidence of stable social aggregations occurring in a whole range of different lizard species. I guess the main group of lizards for which that have kind of championed this kind of push to accept lizards as model systems for studying complex social evolution has been an Australasian group of lizards, the Egernia group, or which is recently renamed the Tiliquini group. This is research that was pretty much started by the late, great Mike Bull and his work on one particular species of Egernia, the sleepy lizard, and his documentation of long-term pair bonding in the sleepy lizard. So his research was the first to show that lizards can form long-term stable pair bonds that can last multiple years. And this really kind of springboarded a kind of exploration of stable social living across the Egernia group more broadly. And Mike and subsequently his students then kind of expanded this research out to other Egernia species. And in these other Egernia species, they showed evidence not just of long-term pair bonding, but also of delayed juvenile dispersal and prolonged association between parents and offspring. So the emergence of small family groups. The kind of 2 or 3 decades since, and kind of further exploration of different members of the group have kind of uncovered almost the full gamut of social diversity in this Egernia group lizards in terms of vertebrate social diversity. So we have species that are largely solitary, like we might typically expect lizards to be. As I said, these sleepy lizards that live in these kind of long-term monogamous pair bonds. Then you have species like these white skinks that I'm going to talk a lot about today, where you also not only get long-term pair bonds, but you get the retention of offspring, resulting in the formation of small family groups. And then in some species, you get even more extreme examples of social organisation emerging. You get species that live in large communal family groups, sometimes consisting of 5 cohorts of offspring, lots of multiple adults co-occurring in these social groups, including potentially non-breeding adults that are potentially be retained in those social groups into adulthood and do not breed. So as I said, potentially the full gamut of social complexity we get within this one group of lizards. Most of our research has focused on one particular species of Egernia, and that's the white skink here, Lyophilus whitei. So the Egernia kind of spans 63 species and 8 different genera. We work on one particular species, Lyophilus whitei, the white skink. We've spent a lot of time with the white skink trying to first up kind of describe what family life looks like in these lizards, but then also kind of quantify variation in family life. And then also use these lizards to kind of try and explore some of the factors that kind of correlate with that variation in family life. So our research on the white skink is kind of based on 2 key empirical resources. The first is a long-term field study. So this is a field study that's been going on for about 20 years now. And in this field study, our aim here is to quantify variation in family life within these lizards. This field site is out on the east coast of Tasmania, so about an hour's drive from the capital Hobart, which is down here. It's in a patch of kind of dry, open woodland. The study site is pretty small. It's about 400 metres by 200 metres. and it contains probably anywhere between 100 and 150 lizards in the early days, although those numbers have decreased over the last couple of years. So this site contains, as I said, about 100, 150 lizards. They're all permanently marked, and each year we go out and we capture our lizards using our highly sophisticated lizard fishing technique, which is, it's pretty much does what it says. You tie a mealworm at the end of a fishing line, you dangle that mealworm in front of the lizard bites on, you lift it up into a bucket, and you've got your lizard that you can then take various measurements on. We also then mark it temporarily with a cloth tape that allows us to identify individuals. And then we spend the entire activity season, which spans late August all the way through the austral summer to early April, following individuals, recording their positions, recording which burrows they're in, and recording any kind of social interactions we observe that they undertake. All up, we've got about 20 years of data. We've been doing this for about 20 years or 21 years this year. And this data set now contains about 400 animals that have been caught or recaught, and about 900 offspring that have been born in the lab. The other resource that we have, and the other really neat thing about the lizards, is that they're amenable to experimental manipulation. So we can use our natural population data to kind of generate hypotheses, or we can identify variation in social behaviour. We can then generate predictions for what might drive that variation in social behaviour, and we can bring other lizards into the lab and set up large-scale experimental manipulations that allow us to test causally the extent to which those predictions play out in controlled experimental contexts. So that includes large-scale experiments where we can do this at the population level, so we can put lots of lizards in these enclosures and manipulate things at the population level, or it includes these kind of smaller enclosures where we can manipulate things at the family level. And I'll talk a little bit later on about how we've utilized these resources to test some predictions about what might drive variation in social organization and family life within these lizards. All right, so just to give you a little bit of an idea about what social life and family life looks like in these lizards, as I said, we spend the entirety of the active season collecting positional data on these lizards. We then use that positional data to generate home range of each lizard in our population, and we use home range overlap to get some idea of the social structure of these lizards. So this is the study site here. We have a highway that goes across the site here, which prevents dispersal from out of the home range, out of the study site, sorry. It also provides opportunities for passerby who see you with a fishing rod dangling some bait into some rocks to stop and provide some constructive feedback as to what on earth you're doing there. The other side of this home study site here is unsuitable habitat that the lizards don't necessarily like to go through. So this is a semi-enclosed population. It's about 400 metres by about 200 metres. And it's got about 80 adults and about 40 to 50 juveniles or sub-adults in a given year. Once we've kind of generated the home ranges, this is an example of 1 year of data here. We've got the home ranges of all adults in the population, and we want to use home range overlap to get some idea of the social structure, the adult social structure of our population. And looking at this figure here, you probably can see that it's a pretty difficult undertaking. It looks like a bit of a mess. If we kind of reduce this down away from the total home range down to the core area home range, we see a different pattern kind of emerging, and one that kind of provides a much clearer picture of the adult social structure. And that looks like this. And what you can see here is across the study site here, you see multiple instances where you get a single black dot, which is a male, and a single white dot occurring right across the study site. So these are examples of where you have a single adult male and you have a single adult female co-occurring together across the duration of the active season, not just in the mating season, but throughout the active season. The location of these dots is really important because they tend to settle on a key feature of Egernia ecology, which is a reliance on either a burrow system or a crevice site. So across the system, we have a bunch of crevice sites or burrow systems that these pairs kind of utilize. And each one of these black and white dots is basically a male and a female sitting in a crevice site or sharing a crevice site, and they do that for the duration of the year. So most of these black and white dots that you see here are a single male and a single female, so an example of social monogamy. And in fact, about 70% of our adult social groups are socially monogamous. When you look in the middle of the study site here, you see a slightly different pattern emerging, where you see a single black dot and multiple white dot. And those are examples of where a male has been able to acquire either multiple burrow systems and multiple females, or has a burrow system that's been able to attract multiple female partners. So even though the majority of these adult social groups are socially monogamous, we still do get a fair bit of social polygamy going on. So about 30% of social groups are socially polygynous. As I said, these social groupings are extremely stable within years, but they're also extremely stable between years. So if you go to the same burrow system the next year, you will find the same pair in that burrow system. So we get about 70 to 80% pair stability from one season to the next. That doesn't mean that things don't change. There is still some dynamics in the system. This is an example of transitions from one year to the next, focusing on 5 different pairs. You can see that most of these are consistent across years, but we do get some variation. So for example here, male 110 has gone from having a single female partner in this year to acquiring an additional burrow system and gaining an additional female partner from one season to the next. Sub-adult 5 here has become an adult, acquired a burrow system and attracted a female partner. So these systems have some variation from year to year, but the underlying kind of pattern is strong pair stability. In terms of pair duration, the maximum pair duration we have is between a male and a female is 9 years. Given that these guys live for about 12 or 13 years, that's almost lifetime pair bonding occurring. That still pales in insignificance compared to some other Egernia species. For example, the sleepy lizards here, the maximum pair duration for a sleepy lizard is 27 years and counting. So these are really strong, stable pair bonds that are kind of emerging between adults in these systems, not just in the whitey-eye, but across other Egernia species. Divorce is relatively rare, so pair separations are relatively rare. When they do occur, they tend to occur because of the death of one individual. Separations where you get both individuals still occurring in the population only occur in about 20% of pairings. So these pairings are extremely stable and very rarely end up in divorce unless one individual dies. And I probably don't need to tell you, but that's a pretty impressive level of pair stability, especially when we compare it to most Western human societies. So obviously that gives us a little bit of idea of the structure of the adult social pairings, but we also want to know something about the consequences of those pairings in terms of reproductive success. To get that information, we go into the field in January at the end of female gestation. These lizards are viviparous. We catch pregnant females, we bring them back into the lab, into our maternity ward, and they give birth to offspring in the lab. And that obviously gives us data on female reproductive output. But obviously it also allows us to get genetic material from the offspring and compare that to the males and the females using paternity analysis to assign paternity to all the males in the population. And this obviously allows us to explore the consequences of that variation pairing behaviour in terms of male and female reproductive success. What does that look like? Well, this again is taking this kind of 1 year as an example. We know that about 30% of females don't reproduce in a given year. So we know that females tend to skip reproduction approximately every 3 years. Of those females that do reproduce, we find that about 70% of females are not only socially monogamous, but they're also genetically monogamous. So all the offspring in that female's litter are sired by her social partner. And there's very low levels of multiple paternity. particularly when in comparison to other lizards who tend to have really high levels of multiple paternity on average. That does leave a certain proportion of offspring that aren't sired by the social female. They're sired by males outside the pair bond, so extra pair offspring, and they tend to be sired by males from closely neighbouring social groups. They kind of wander into the female's social group and territory and acquire a mating with that female. Males gain reproductive success both through acquiring additional partners. The main way in which they can do this is acquiring multiple social partners. So the best thing for a male to do is to be socially polygynous. However, obviously extra pair copulations also contribute to a male's reproductive success. So male can gain increased reproductive success both by increasing the number of social partners he has, but also by acquiring extra pair partners. Females also seem to gain from extra pair paternity through being able to trade up from their social partner in terms of relatedness. There appears to be some constraints on female mate choice in terms of relatedness, so that females tend to pair up with males that are more related to them than an average male in the population. But females kind of alleviate this constraint by mating with extra pair partners that are less related to them than their social partners. Okay, so that's the adult component of these family groups, generally kind of comprised of social pair bonds, socially monogamous pair bonds with some social polygyny occurring. The other key component of Egernia family life is the prolonged associations between parents and offspring. So offspring delaying dispersal and occurring within the parent home range, usually for a year, sometimes for multiple years. This is kind of what it looks like in a given season. What we have here is our adult pair bonds again, and we have overlaid on top of that is the surviving offspring that have kind of been born in January. They've survived through the winter, and we're now recapturing them the following spring. So these are about 6 months old. This is about 40% of the offspring that are born, so survival is about 30 to 40%. And you can see here the green dots are offspring when we go and recapture them are recaught in their parents' home range. So in the burrow system, sharing the burrow system with their parents, whereas the red dots are individuals that have dispersed out of their parental home range and have set up an established territory somewhere else in the periphery of their parents' home range or sometimes elsewhere on the study site. So offspring will stay in their parental home range usually for a year, but occasionally they will be retained in their parents' home range for a couple of years, and they don't mature until they're about 3 years old. So once they've kind of gone to 2 years, they'll then go off, disperse, and start to establish their own territory as they transition into adulthood. One of the really striking things about the extent of parent-offspring association here is that there's huge variation across years, both in terms of the number of social groups that tolerate juveniles, but also that tolerate subadults. So this is kind of quantifying that across 10 years of data. And what you can see here, first up, is that a lot more social groups tolerate juveniles compared to subadults, although in some years there's more subadults than juveniles in social groups. But most importantly is that there's massive variation in the extent of parent-offspring tolerance from year to year. In some years, almost every social group, you have offspring being tolerated. In other years, almost no social groups tolerate offspring. So obviously there's huge variation here, and we kind of want to get a handle on what factors drive that variation in parent-offspring association from year to year. And I'm going to talk a little bit later on some experiments that have kind of explored some of the mechanisms that might be responsible for this variation. So we have parents and offspring associating with one another, and this leads to obvious questions about the potential function of that association and to the question of whether this represents some form of parental care. We would suggest that it does for a number of different reasons, and this is because the presence of an offspring or the tolerance of offspring by adults provides various benefits to offspring. The main one being is it reduces aggression towards the offspring from hungry conspecifics. Conspecifics are a key source of mortality to the offspring through infanticide, and having a parent around almost eliminates that risk. This is evidence not necessarily from, was not from the white skink, but this is from a closely related black rock skink, Egernia saxatilis. And this is a relatively simple experiment where Dave O'Connor and Rick Shine looked at the amount of aggression directed towards an offspring by conspecifics in the presence versus the absence of their mother. And you can see here that when mum is absent, there's lots of aggression directed towards offspring, that you place mum there and you almost completely eliminate that aggression. We also know that female aggression increases during the reproductive period, so it ramps up as you go throughout gestation and is maintained at high levels into the postpartum period when offspring are presumably most at risk. So females have heightened aggression during the early periods of postnatal care. We know that consistent individual variation in aggression of female aggression correlates quite strongly with variation in offspring survival. So offspring from mums that are more aggressive have increased survival compared to offspring from mums that are less aggressive. So all of this is pointing towards a real advantage of being tolerated by mum in terms of protection from conspecifics and conspecific infanticide. There are several other benefits that offspring gain from being associated with mum or being tolerated with mum. One is that it provides an opportunity for offspring to display a range of behavioural traits that may have other functional benefits in terms of ability to kind of acquire resources. So this is this is an experiment run by Kirk Munch during her PhD, and she brought mums into the lab. She allowed them to give birth, and then she held offspring either on their own or with mum during the early stages of the postpartum period, so the 6 weeks following birth, and she quantified a range of behavioural traits and compared behaviour of offspring when they're with mum versus when they're alone. And she found that offspring with mum displayed a whole range of behavioural traits, increased boldness, increased exploration, and increased activity compared to offspring that were on their own. And this may have really important functional consequences for their offspring, because it suggests that offspring, when mum is around, display these behavioural traits that may allow them to acquire resources and potentially have benefits in terms of increased growth. And mum's not doing anything in these contexts, she's just sitting there, but the presence of mum obviously elicits this kind of behaviourally plastic response in the offspring that potentially allows them to acquire a greater amount of resources. And in line with that, when we kind of have a look at some of the growth benefits of being tolerated by mum, both in the field and in outdoor enclosures, we find that offspring that are tolerated by mum tend to grow more than offspring that are not with their parents. So on a whole, a number of kind of relatively subtle benefits of being tolerated by mum. Another kind of important context or important context with regards to these kind of parent-offspring associations is that there doesn't seem to be any costs associated with these parental tolerance. So, which is pretty not necessarily surprising, because mum is not really doing anything. She's just sitting there, the offspring, but her presence kind of reduces the threats to the offspring and provides offspring the opportunity to elicit this kind of behavioral plasticity. She doesn't pay any costs. And this might be really important when we kind of think about how parental care kind of initially evolves, is that during the early stages, you kind of need a low level of costs to the parents and these kind of subtle benefits to the offspring. And that might be really important for getting these systems going in terms of stabilizing these associations between parents and offspring and providing a context from which you might be able to get the evolution of more elaborate forms of parental care. So that's what parental care looks like. In family living looks like in these lizards. It's based primarily on long-term stable monogamous pair bonds between males and females, delayed offspring dispersal, resulting in the emergence of these small nuclear family groups. This is relatively consistent with a range of other Egernia species that also display these relatively simple nuclear family groups where you have males and females in stable relationships and usually one cohort of offspring. Species like black rock skinks, tree skinks here, the land mullet all display these relatively simple forms of small nuclear family groups. And when we kind of expand out across the Egernia group more broadly, we know that there is greater variation in social organisation, including a range of species that exhibit that are largely solitary. So species like Carisha zebrata, Taliqua adelaidensis, Lysolepis coventrii. These guys don't display any long-term pair bonding or any kind of prolonged parental care beyond, the first few hours or maybe days of life of the offspring. We know that there are other species that also exhibit no parental care but long-term pair bonding. And then there are species such as the Gidgee skink and the Cunningham skink, where these kind of simple forms of family life have kind of extended into large communal family groups where you don't just get one cohort of offspring remaining with mum and dad, you get multiple cohorts of offspring, including offspring that potentially persist in those social groups into adulthood and delay their breeding and their dispersal. Okay, so we've kind of described a lot of the patterns of family life in both the white skink and the Egernia group more broadly. Now we're going to shift a little bit to the processes that may have kind of facilitated the emergence and elaboration of social life, family life in these skinks. So here we're kind of going to focus not so much on the pair bonding, but we're going to focus on the prolonged parent-offspring associations. So we're going to focus here on how we kind of get transitions from these solitary ancestors where offspring disperse immediately upon birth or hatching to the emergence of these small, simple social groups, and how those set the foundation from which you might get these more elaborate communal social groups through the retention of multiple cohorts of offspring. We're also going to think a little bit about how those social groups, once they kind of stabilize, as I said, set the foundation from which you get selection on more complex social behaviors, so more complex forms of parental protection, potentially parental provisioning, and increased complexity in kind of communication. Okay, so what are the kind of factors that might have influenced the kind of emergence of these simple social groups, so have facilitated the prolonged retention of offspring with their parents? One factor that we thought was going to be particularly important in this context was one key life history trait, which is live bearing. So lizards have two different parity modes. They can both give birth to live young, but they can also lay eggs. When we kind of have a look at the distribution of parent-offspring associations and social groupings in lizards, across lizards, we find that they're biased heavily towards live-bearing species. And we're interested in understanding whether that kind of plays out at kind of macro-evolutionary scale. So why might live bearing promote the evolution of social grouping over and above egg laying? Well, the simple kind of explanation is that live bearing potentially creates greater opportunities for environmental factors to influence or bring together parents and offspring. So to set the stage for the early emergence of parent-offspring associations. So it works a little bit like this. So if we have an egg-laying oviparous reptile here that's laid its eggs, there's often a long period of time between the production of the eggs, the laying of the eggs, and when the offspring are going to be produced. This can be a number of days, it can be a number of weeks, it can be a number of months. And during that time, there's lots of opportunity for the female to disperse away from the egg-laying site. And that, as a consequence, the female is not necessarily going to be present at the production of the offspring. If you contrast that to a viviparous species, where embryos are gestated within the female, when offspring are produced, the female is always going to be associated with the offspring, at least at the initial stages of offspring production. And what that might do is simply create greater opportunities for environmental factors to basically keep parents and offspring together. So if environmental factors allow dispersal, then offspring can disperse. But if there's some kind of restriction on dispersal that keeps offspring at the natal site, this is more likely to create an association between parents and offspring in a viviparous species compared to an oviparous species where the female may have dispersed well away from the nest site. So we wanted to test this hypothesis. We can't obviously do this at the individual level because individuals don't vary in parity mode. The large variation that we see in parity mode is across species. So what we wanted to do is test this at a macro-evolutionary scale. So this was work done by Ben Halliwell, who was a PhD student. And what Ben did was went and collated information on social grouping, particularly focusing on parent-offspring associations across all squamate reptiles, so lizards and snakes. So he collected data on about 1,200 different species, 100 of which we know exhibit, or he could confirm exhibit social grouping. He then mapped those across the squamate phylogeny, showing that social grouping has evolved multiple times independently across both lizards and snakes. He could then compare that to the reconstructed evolutionary history of viviparity, which also shows considerable lability, and he can compare those evolutionary histories and see how they kind of how similar they were. And obviously what you can see here just by eyeballing this data is that there's quite a neat association between the emergence of viviparity and the emergence of social grouping. So a nice evolutionary correlation between this key life history trait, viviparity, and the emergence of social grouping. Now, this is not to say, obviously, that social grouping can't emerge in oviparous species. It certainly can, particularly in territorial oviparous species that are highly that kind of stay with their at their nest site. You know, that's another route to social grouping. But certainly, there's overwhelming evidence here that social grouping has evolved more readily from a background of viviparity than it has from a background of oviparity. This is obviously not, viviparity increases the opportunity for social grouping. It doesn't necessarily make social grouping occur. There you need other environmental factors that keep parents and offspring together, that restrict dispersal of the offspring. So the next step in kind of exploring what factors have influenced the evolution of family life in these lizards to try and understand some of the environmental factors that have kept parents and offspring together. And this is where our large experimental facilities really come into their own, because we can generate hypotheses about which environmental factors might be particularly important. And then we can manipulate those at a large kind of population level and have a look at whether they do initiate a greater level of association between parents and offspring. In the Egernia lizards, as with many other organisms that delay dispersal, habitat constraints is probably the key factor that is likely to be responsible for delayed dispersal. As I mentioned before, Egernia lizards rely on burrow systems or crevice sites, which are patchily distributed across the environment. And this probably is a key constraint to dispersal of offspring and maybe initiate greater level of association between parents and offspring. So we wanted to test this idea using our white skink. To achieve that, we created 2 different experimental treatments. We brought female lizards into the lab, we let them give birth, and then we released them into these outdoor enclosures. These enclosures were made up of 2 different components: a home enclosure where we released the mothers and the offspring, and a dispersal enclosure. Only offspring could pass from one side of the enclosure to the other. And in the dispersal side, we created 2 different treatments where we manipulated the level of habitat saturation by altering the density of adults. So in one treatment, we created a low saturated environment. So we only released 2 adults, leaving plenty of habitat available for offspring to disperse into. In the other treatment, we released 4 adults, which saturated the habitat and reduced the amount of available habitat for the offspring to disperse into. We then followed the offspring for 8 weeks, recording their dispersal patterns, where they settled, and whether they settled on back with their mum. And what we found was quite clear evidence that habitat saturation and environmental ecological constraints certainly play a key role in mediating the level of parent-offspring associations in these lizards. So we found, and we compared to the offspring exploration behaviour, we found that offspring tended, but in both the high and low saturation treatment, tended to explore the dispersal treatment to the same degree. So they both moved around and explored that dispersal treatment, but a far greater number of offspring settled in that dispersal treatment in this low saturated treatment, where there was a lower number of adults, more available habitat, compared to the high saturated treatment. And the consequence of that was that when we look at the amount of parent-offspring associations, there was almost twice as many parent-offspring associations occurring in that high saturated habitat. So when we restricted the opportunities for offspring to disperse, offspring tended to stay with their mother, leading to a far greater level of parent-offspring association compared to when they had opportunities to disperse, because the habitat wasn't as saturated. So we've got viviparity as an important precursor. That creates a greater opportunity for parents and offspring to become associated, because it creates an association between particularly mother and offspring at birth. And then we have ecological factors that basically restrict dispersal of those offspring and keep those offspring, particularly with mum, but if you have a pair bonding species, also with dad. Okay, but it's not kind of an all or nothing scenario. We still have variation in the extent to which habitat constraints, for example, create associations. There's still plenty of examples of where habitat constraints don't create these associations. So the next kind of question is, what explains this variation in this plastic response to habitat constraints? And one explanation might be that this is driven by fundamental differences, intrinsic differences between the females in how tolerant they are of the offspring. So it might be that even though habitat constraints restrict offspring dispersal, it might be that whether the offspring stay or not are kind of dependent on aspects of the female behaviour and how good she is at actually tolerating those offspring. We've kind of got some, this is data from Victoria Russell's PhD that kind of explored the extent of variation in tolerance and social tolerance across a whole range of different Egernia species. So she brought mums into the lab, she let them give birth, she just kept them in close association with their offspring, and then she recorded various behavioural responses of mothers to their offspring across a range of different social axes to look if she could quantify variation in social sociability or social competence across females. And what she found was, across a range of different species, she found quite strong evidence that you get huge variation between females in how tolerant they are of the offspring. Some females are super tolerant across multiple sociability axes, so they're consistently tolerant of their offspring. They're always with their offspring. Whereas other females in the population show extremely low levels of tolerance. They're hardly ever with their offspring. They're quite often aggressive towards their offspring, and in some instances, this can even result in infanticide. And we know from a range of Egernia species that if you keep females and offspring in the lab, there's a certain proportion of conflict that goes on between, well, certain females that are aggressive towards their offspring, and a certain females that will, in fact, even consume their offspring. So within a population, there's huge variation amongst females in how tolerant they are of offspring, and this may dictate which of those females tolerate their offspring when habitat constraints kind of restrict offspring dispersal. I just want to kind of touch a little bit more on this kind of infanticide risk by the females and the importance of kind of getting over this risk of infanticide for getting these kind of parent-offspring associations started. This is some recent work that we've done with a PhD student that's just handed in, Jai Lake, and a collaborator, Dustin Rubenstein, that I'm sure everybody's aware of. This is an experiment where we brought females into the lab. We kept them either with their offspring or without their offspring, so it effectively created caring and non-caring females. We kept them like that for an 8-week period of time. We then took the offspring away from the caring females for a 12-hour period, and then we either dropped that offspring or the offspring that had been kept from this female for 8 weeks back into the female's home range behind a plastic barrier, or we dropped a control object and then we looked at the female's behavioural responses to these objects to get an understanding of how females responded to offspring, both when they'd been caring for their offspring and when they hadn't. These are kind of the behavioural responses that we found from that experiment. And what we found was that females, whether they had been isolated or with their offspring, exhibited responded to the offspring to a much stronger degree than they responded to the control object. So they tongue flicked the offspring much more, and they also performed a lot more lunges towards the barrier and towards the offspring compared to the control object. So they're clearly recognizing the offspring compared to the control object. And these responses were much stronger from the female that had been isolated for 8 weeks compared to the female that had been with the offspring, potentially because the female with the offspring could recognize the offspring as its own, whereas this female that had been isolated couldn't. The other thing we did then, 45 minutes after we kind of exposed them to this kind of stimulus, we then sacrificed the females. We extracted the brain, we sectioned the brain, we punched it for 6 brain regions that we know are important in the social decision-making network. We then did RNA-seq on those brain regions and looked for differential gene expression between these 4 different social contexts. What we found was that there was a relatively low number of genes that were differentially expressed across most of our brain regions, but there was one brain region that appeared to elicit some particularly interesting results, and that was the paraventricular nucleus. There we found 4 key genes that were differentially expressed, mostly when you dropped the offspring in compared to the control object. These 4 genes were GAL, PMCH, and TAXC, and all 3 of these genes have been associated with the initiation of feeding. And this is potentially really interesting because it suggests that when you drop this offspring into the mother, the first response that the mother has is a feeding response. So it's the initiation potentially of a desire to eat its own offspring. The only other gene that showed differential expression was a gene called CARP-PT, and that's been involved in feeding suppression. And this gene might be really important in kind of restricting or stopping females eating their offspring. And that may be kind of an important step to get these parent-offspring associations going. The first step being simply do not eat your offspring. Now, there's a fair bit of water to go under the bridge in terms of these results moving forward, but we think this is a really kind of exciting potential way forward because it suggests that obviously in order to kind of evolve or kind of stabilize these parent-offspring associations, you need to kind of suppress the urge to eat your offspring. So I've talked a fair bit about the role that females play and variation in female behavior and aggression might play in allowing offspring to be tolerated within the home range. But obviously that's only one social interaction. There are other social interactions that are going to mediate family dynamics. For example, the role that males potentially play, the father plays. It's much more difficult to kind of do experiments on the males compared to the females because we need to know which male is the father of the offspring. So it's a little bit harder to kind of do those experiments. So we have much poorer understanding of the role that males play in all this. But the other entity is obviously the siblings, because once females are tolerating multiple siblings, those siblings themselves can kind of interact. And what we think probably goes on here is that female tolerating offspring are really important for dictating whether offspring remain within the parental home range per se, but then the dynamics of who stays and how many offspring stay are probably dictated by interactions between the siblings, because the siblings in about 50% of litters really do not like each other. This is kind of a typical day in the life of lizard siblings. And so it's pretty much constant aggression. In about 50% of the litters, it's pretty much constant aggression between the siblings. One sibling in particular will pick on the other sibling, and sometimes the kind of conclusion of that is reduced growth to start off with, but usually mortality. Now, obviously, this is in a lab scenario. In the field, you can imagine that probably results in dispersal of the dominated sibling. And we know that compared to mother offspring aggression, there's lots more sibling aggression going on. So sibling conflict is potentially a really important kind of mechanism that influences family dynamics once you've got parents tolerating offspring. So mothers don't really get involved in these interactions between the siblings. They just sit there and let it happen. But they don't directly get involved in the conflict, but they can influence the conflict in other ways. And one kind of interesting way that they can influence the conflict is through a process called birthing asynchrony. So one other unique thing about Egernia lizards is that unlike almost all other reptiles that we know of, Egernia lizards give birth to offspring one at a time. So even though offspring are fully developed and ready to go, they will give birth to one offspring and then hold on to the remaining offspring, sometimes for a day, sometimes for a week, sometimes for 2 weeks before they give birth to the next offspring. And this creates really large competitive and dominance hierarchies between the firstborn offspring and subsequent born offspring that might play important roles in mediating the competitive environment within these broods and ultimately dictate which offspring disperses and which offspring remains. And this is obviously analogous to what we see in many bird systems where you have hatching asynchrony. We can kind of test the role that asynchrony plays in mediating conflict and mediating the emergence of these dominance hierarchies, because we can experimentally induce asynchrony by pumping females full of the birth hormone arginine vasotocin, forcing them to give birth to all offspring at the same time, and then create experimentally asynchronous and synchronous broods, and then look at what that means for the emergence of these size hierarchies. And that data suggests that when you create asynchronous broods, you get one offspring becoming dominant. It's usually the firstborn offspring. That offspring grows much better. It has increased survival compared to its siblings, and it also spends more time with its mother. We also know that the extent of birth spread, so the amount of time between birth in days, can dictate the amount of conflict that occurs. So the extent to which these dominant hierarchies kind of emerge depends on the extent of birth spread. So when you have births that are close together, you tend to get lots of conflict occurring and lots of mortality of second and third born offspring. But if females spread that birth out, those hierarchies are already pretty much established, and you see much less aggressive acts, and you see greater levels of survival of the later born offspring. The final kind of interesting point relating to this is that there's only one other group of lizards that we know have this birthing asynchrony, and that's caudilid lizards. And the other main interesting thing about caudilid lizards are that they also exhibit family living. They're the other kind of major group of lizards that we know exhibit family life. So we have this kind of convergent evolution of family life that is associated with this convergent evolution of birthing asynchrony, which probably suggests that birthing asynchrony may have evolved as a way to moderate conflict within these litters. Okay, so I just got a couple more things I want to talk about before I finish up. So we've kind of got the emergence of these simple forms of family life where you get this prolonged associations between parents and offspring are underpinned primarily by the fact that these lizards are viviparous, by the fact that they rely on particular ecological factors that are restricted in the environment. So you have ecological constraints. They're probably also influenced by the variation in female propensity to tolerate offspring, how good they are at tolerating offspring versus not, I guess, consuming offspring. And the dynamics are probably mediated to some degree by sibling conflict. Ultimately, once you have the stabilization of these social groups, so if environmental conditions that produce these social groups become recurrent, and there are benefits to living in these social groups, so you get selection on variation in various traits that individuals kind of exhibit that kind of allow you to stabilize these social groups, then this obviously sets the stage from which you can get kind of get the emergence of more complex forms of social organization. So transitions from a single cohort of offspring to the retention of offspring for prolonged periods of time and the build-up of multi-cohort, multi-generational family groups. But as I mentioned at the start, it probably also sets the stage from where you could get the emergence of more complex forms of social behaviour. So more complex forms of parental protection and potentially the opportunity for things like parental provisioning to emerge. So just a couple of final videos to show examples of this. The first one, we've talked a little bit about how parental protection or tolerance of offspring provides benefits in terms of reduced conspecific aggression, and that protection is relatively indirect. It's just the presence of a mum. But obviously, as these groups stabilise, you might expect that protection becomes more direct, and it may even kind of transition into not only protection against conspecific, but also protection against other threats. And I'm just going to show you a video from one of the more, this is from Egernia cunninghamii, which is one of these species that exhibits these really elaborate and large communal family groups. Oops, get this started. So what we have here is a mother here, and we've got 3 offspring living in this rock crevice here. So the mother is there, you'll see her move, and these 3 offspring. And this is an example of mum protecting the offspring, not necessarily from a conspecific, but from another threat. She will move in a minute. I promise. Now that's mum that's seen a threat in the context of a large brown snake, and she's left her 3 offspring that have disappeared off to the crevice, and she's gone over, grabbed that brown snake and sent it away. This is a very brief, similar kind of footage of the female doing the same thing to a magpie. So in those instances where we have these kind of more complex social organizations, social groups kind of emerging that are kind of, I guess, further along the transition to more complex forms of sociality, we potentially also see the refinement of more complex forms of social behavior and parental protection. And the final kind of video I want you to show is the same kind of idea, but in the context of parental provisioning. So this is something we see relatively commonly in the lab, and that's mums eating mealworms and then allowing offspring to consume food from her mouth. Not necessarily, she doesn't seem particularly happy about that, but nevertheless is tolerating it. And off the offspring goes. Now, I'm obviously not for a moment suggesting that is parental provisioning in any shape or form, but what I want to suggest is that once you have these kind of simple associations between parents and offspring occurring, then instances like this, where females tolerate offspring sharing food, may form the foundation from which more complex forms of parental care, such as parental provisioning, can occur. So if there's variation between females in how likely they are to let offspring take food from their mouth, And under environmental conditions where food is in short supply, you can imagine a scenario where that can form the foundation from which you get more complex forms of parental care, such as parental provisioning. Okay, so we've kind of talked a lot about, we've defined what social living and family life looks like. We've explained that it's relatively simple, expanding simply to prolonged associations between males and females, delayed offspring dispersal, and prolonged associations between parents and offspring. We've kind of talked about the fact that these things can emerge relatively simply. There are a few predispositions and exposure to environmental constraints. And as long as you have females that are willing to tolerate their offspring, you can get the build-up of these social groups relatively easily, which leads us to the question of, well, if it's so easy, why aren't there more social lizards? And I guess the answer to that question is, well, there perhaps are, we just need to look. We already know, as I said, as I showed from Ben's data, that social grouping has emerged, probably over 20 times independently across lizards and snakes. We know that there's over 100 different species that exhibit simple forms of parent-offspring association. These are some of the better examples of that, the more well-known. So the cordylids, the xanthusids in America, the oligosomid skinks in New Zealand, the phynocephalus in China, and the lyolamids in South America. But I think if we go out there and look, we'll probably find more examples of these simple forms of parent-offspring association. These guys don't display overt social behaviour, which probably provides an explanation for why we miss a lot of this behaviour. We see adults and offspring together. We very rarely think it looks like a family group. So we probably want to go out there and look a little bit more carefully to see if we can describe more examples of these simple forms of social life across the lizard phylogeny. And one of the real benefits that lizards and squamates more broadly provide is that we already know that social grouping has evolved multiple times independently. So this provides real power for comparative analyses to look at the convergent evolution of social grouping from both a kind of mechanistic but also functional perspective. So more broadly than that, I think lizards potentially provide a whole heap of additional opportunities, as well as the fact that, we've got lots of independent transitions. That is that they're amenable to experimental approaches. So this means that we can kind of study the ecology of social living through targeted experiments and long-term field studies. We can kind of delve into the mechanisms of social behaviour, so the role that genes and development play in mediating variation, in underpinning variation in social behaviour. We've got diversity within groups that allow us to kind of take predictions from our ecological studies and look at how those predictions play out across whole groups, across deep time. And we can even ramp that up across to lizards more broadly, kind of look at independent transitions in a kind of comparative context. So hopefully what that means is that when we kind of talk about model systems for study social evolution in the future, we can kind of place lizards right up there with some of these more traditional model systems in terms of birds, mammals, social insects, fish, and amphibians. And with that, I'll finish up and ask for any questions.
Yes, thank you very much, Geoff. Before we come to the discussion, I would like to. Can you stop sharing your screen? I don't see how I do
Yeah,
this.
sure.
I would quickly like to.
How do I stop sharing?
To announce the next finds that are going to happen, also because I see that there's in the audience some people that are not so common here. That's the season find in fall with lots of interesting talks. If you're not subscribed yet, send us an email to this email that you want to be in the list, and you will be informed about everything. And otherwise, we will also announce it on social media soon and send it to the finalist latest in August. Yes, so thank you very much for a great talk. Also about the questions, if you have a questions, please type a question mark into the chat. And if you're a student, you can put an S behind it, then we will preferably refer to your question. If you have a direct follow-up questions, you can write FU in it. It's already a bit late for everyone except for Geoff. For him, it's very early because it's 20 past 2 in the morning. But still, so that he can still get some sleep. And when you ask a question, please try to be very precise and short so that we have enough time to deal with a lot of questions and don't get too late, too early for everyone. Okay, so how do I stop this sharing? And this every version is okay. Are there any questions? I don't see anything yet. Then maybe I can start. I have a lot of questions.
I think
Geoff,
Sarah might have a question.
oh, Sarah, yeah, she has the hand up. Okay, Sarah, you can start. You're muted. How do I get this?
Apologies, I can't make the system work, but thank you. That was wonderful. I was fascinated by your finding that for care to begin, you had to suppress the cannibalistic possibilities first. And given what people like Catherine Dulac and Lauren O'Connell are finding, did you look at galanin and at some of the neuropeptides and hormones involved and what's going? Tell us more.
Yeah, so that those results, I guess, were kind of exploratory in a sense. So we kind of set up this experiment, and it was whole RNA-seq. So you know, the results that we found in terms of the feeding suppression and the feeding initiation were quite surprising in a sense, but we didn't kind of necessarily target them. The interesting thing, I guess, is that we've focused on some of the neuropeptides with Dustin and Martin, mainly on oxytocin and arginine vasotocin as potential mechanisms for social bonding, underpinning social bonding. So we've actually done, and I didn't have time to talk about a lot of stuff, but we've actually done a range of experiments where we've kind of used pharmacological manipulations, focusing on oxytocin, and to try and get a handle on whether oxytocin, like in other systems, kind of underpins variation in pair bonding. We find some kind of subtle evidence of this in both the white skink, but also in one of the Taliqua species, but nothing to kind of really write home about. And more recently, we've kind of been thinking about, during these early stages of social evolution, whether those kind of affiliative hormones are less important, and it's more about the kind of restriction of the aggressive hormones.
Yeah.
So to get these things started, maybe you need to avoid doing these angry things and aggressive things. And then once you've kind of stabilized these systems, perhaps that's when you kind of get these more affiliative mechanisms playing a role. And that was why these kind of the kind of genes that kind of popped up, and as I said, it's still early days, were kind of supportive of that, right? Because it really, and we see it in the lab, it's not unusual for a female to go rogue on her offspring. And that seems to be part of the natural variation that we see in the population. So in the population, we have natural variation in female parental care ability. Some females are very poor at it. Some females are very good at it. And I think probably, stabilizing this system needs to get over that hump of, the initial kind of response of eat what's in front of me. It's a little tasty morsel. That needs to be a kind of a key component to this. And one kind of thing that I've kind of thought about in this context relates back to that association between viviparity and parental care. And we kind of explain that association in that it simply increases the opportunity, right? Like mum's there, she produces offspring. So any environmental factor that restricts dispersal creates this association almost by definition. But there are a number of other mechanisms that might be important in the context of viviparity. And one thing that is different between a viviparous species and an oviparous species is that oviparous species potentially don't have the problem of eating your offspring as soon as they're produced if mum's not there when they hatch, whereas viviparous species perhaps do. It might only be for a couple of hours, until the offspring disperse, but they certainly don't want to be munching down on the offspring as soon as they're kind of produced. So perhaps transitions to viviparity have had to kind of solve this problem, at least in the short term, of not eating offspring. And maybe then in these social systems where you get these prolonged retention of offspring, you just kind of prolong that mechanism, maybe through feedback loops with associations with offspring, so that you don't eat your offspring, weeks later, because they're still around you, if that makes sense.
Did you look at any differences in the monogamous pairs between the male and the female in this?
No. So one thing that we haven't got a handle on is what the males do in the male role in all this, because obviously the male's there as well, and the male could just as easily show the same variation. Yeah, exactly. So the males we haven't really focused on, but certainly they're an important part of this, because it might be not just that mum doesn't eat the offspring, it's also that dad doesn't eat the offspring. And so, when you get these habitat constraints kind of resulting in the formation of family groups, maybe you need both mum and dad to be cooperating in terms of their not eating the offspring. So yeah, and the interesting thing is if you mum will care for the offspring and won't eat them, but if you take the offspring away from mum for 2 weeks and put them back, mum's forgotten all about them, and she'll attack them. So there's obviously something going on that creates the kind of reinforcement of this association and potentially these mechanisms that suppress feeding, that if you take the offspring away, you kind of remove that, and then mum goes back to her kind of ancestral roots of eat what's in front of me.
Got it. Thank you so much. Thank you.
Okay, so we have, I believe, 2 student questions, which I will call them up first. The first student question comes from Chuan. I assume at least from that you are a student. Yes, Chuan. Ceballos S. Samuel, can you ask your question?
Hi, everybody. The first amazing presentation. Thank you for that. And the first question is, I don't understand well why parental cares do not generate costs for the parental individuals.
So I think in this instance, it's just because the mum really doesn't do anything, and the dad. So they pretty much just tolerate the offspring. And you could see it a little bit in that video where the parents where the offspring were kind of just going at one another. The mum just sits back, and even when you kind of sit there and watch them in the lab, the mum does her own thing. She certainly, if you drop food into the offspring, the mum doesn't wait to feed. She hoovers up as much as she can, and the offspring are left to what they can get. And sometimes that, as I showed in a few cases, that they get that out of the mum's mouth. But yeah, really in these instances, mum is just tolerating offspring. So this initial step to the emergence of more complex parental care where costs are involved is simply, not tolerating offspring and not eating them. Now, there may be some subtle costs. So if you've got a bunch of offspring in your home range, perhaps you get harassed by other individuals in the population who see those offspring as a food source, but we don't see any necessarily evidence of that. And when we kind of look at any kind of changes in female condition or female scarring or anything like that, we don't see any evidence that mums that tolerate their offspring seem to kind of suffer any of those costs more so than mums that don't tolerate their offspring. And I think that's because we're at the very early stages of parental care in this group, where parental care is so simple. It's just tolerate your offspring, don't eat your offspring. But importantly, that sets the stage. Once you have that stabilized, that sets the stage from which you can get more complex forms of parental care. So in something like the Cunningham skink that I showed at the end, which was fighting off snakes and magpies, obviously, if it's doing that to directly to protect its offspring, then that comes with a kind of considerable cost potential in terms of mortality. But we don't necessarily see any of that in the context of the white skink, and even in the context of the Cunningham skink fighting off the snake and the magpie. We can't really tell in those videos whether it's doing that to protect the offspring or it's just doing that to protect itself, right? But again, either way, the offspring gain, right? It just being in the present. Doesn't matter whether mum's protecting herself or protecting the offspring gain from being in the presence of mum in that context. And that as well as the kind of low costs that mum pays, if she's just doing it not to protect the offspring, may be important for stabilizing these associations during the early stages of parental care evolution.
I do have another question, sorry.
Go for it.
I mean, to be clear, I understood that this parental care and like this kin selection is the main driver for all these social groups to form, right? But is there any other like behavior such as direct reciprocity or something like that between all these social groups or something like that?
So, yeah, so I mean, I would argue, and I didn't have time to talk about this, but I would argue that in these systems, I don't know how important kin selection per se is in these contexts. So these are certainly social groups made up of closely related individuals, but that is probably a consequence of the mechanisms that produce these groups as much as it is a consequence of them being closely related per se. So if you have a viviparous species that's producing individuals, those individuals are by definition going to be related to you. Habitat constraints are going to constrain related individuals around you. Recognition is probably largely a result of familiarity, and the individuals you become familiar with are probably going to be closely related individuals. So the fact that they're kind of closely related groups probably isn't because kin selection has played a role in stabilizing those groups. It's probably because the mechanisms that produce those groups are biasing the formation of these groups towards closely related individuals. So during the early stages, it's just a consequence that they're closely related. As these behaviors, as things like parental care and other social behaviors evolve to become more complex and there are more costs and more benefits, then you would imagine that it might become important that they are closely related individuals. So kin selection might be involved in the stabilization of these social groups. But during these early stages, it may simply be a consequence. And in fact, we conducted an experiment to test this idea because we were like, okay, well, could you create the same level of parent-offspring association if you basically dropped unrelated individuals in with a parent and restricted habitat? Could you get the same level of a social association? And the answer to that question is yes, but not quite to the same extent. So we conducted a large experiment where we had a bunch of females where we released them with their offspring in those enclosures. We had a bunch of females where we released them with different offspring. And then we did the same kind of a bunch of social network stuff to look at the strength of association between parents and offspring. And we found that there was certainly, when we released non-kin, examples of parents and offspring that had just as strong relationships as when we had real parents. But on average, the strength of association was slightly stronger when it was the actual parents were involved. So it suggests that it is possible to generate these social groups amongst non-kin, and we know that about anywhere from 10 to 20% of social groups will have an unrelated offspring within the social group. So they do occur in the wild, and they probably occur simply because the recognition mechanisms are relatively unrefined, and so offspring that accidentally find themselves in these social groups at the right time can be tolerated in those social groups because, those social groups, the mechanisms there to kind of recognize kin may be slightly unrefined during these early stages of social evolution. I don't know if that makes sense.
Thank you.
Thank you so much.
Next question the next two student questions come from the Sakolenko Research Station, first by Perfect and then by Nukulo Lego.
Hello everyone. My name is Perfect and I'm the research manager at the Circular and Karoo Research Station. I'm currently working on the project to study the activity budget of the lodge building bush Karoo rat using camera traps. So, that was a massive talk, by the way. Thank you very much. I have a lot of questions, but I'm just gonna ask two of them now. So the first question is that based on your findings, like is there a tipping point or a threshold beyond which the benefits of the family living begin to decline, you know? Then the second one would be in what ways does the social organization of the species of lizard parallel or diverge from that seen in birds and other mammals with the same complex family systems?
Yeah. So, your first question is, at what point is there a tipping point where social family life becomes not beneficial, I guess? Is that the question?
Yes, the benefits becomes to decline.
Uh, I guess we don't really have a handle on that. So, well, so when you kind of map social organization across the Egernia group, and there's a lot of information on some species of Egernia, but poor information on other species. So this is kind of pilot data. We think that there's probably a number of species that have certainly lost sociality. So they've transitioned back from living in these kind of small nuclear families to being largely solitary. And in fact, that species that exhibits the long-term pair bonds but doesn't have any evidence of prolonged parent offspring associations, I think is probably an example of a species that has lost parental care. And I think that in that instance, it's not so much about the costs of parental care or family living became so great. I think that species moved into a habitat, so it became less reliant on rocky crevices and burrows and moved into kind of almost a grass or shrubland where the availability of resources was much greater, and offspring simply started to disperse rather than hang around with mum and dad. So there's no evidence that there's costs have got to a point where you get a selection against social grouping, but there is evidence of the loss of social grouping, or perhaps evidence of the loss of social grouping, but I think it's probably a relaxation of the mechanisms that produce social grouping in the first place, if that makes sense.
Yes, it does. Yes.
And in response to your second question, which is how does this compare to social living in other mammal groups or other kind of social vertebrates? So I would argue that we see almost all of the diversity in social living in the Egernia, right up to, but maybe not kind of expanding to cooperative breeding. So we get those large communal family groups that have multiple cohorts of offspring. They have adults that have potentially delayed dispersal and been retained within the social group that don't breed. We don't have evidence that they cooperate, but also in these lizards, we've got no evidence of provisioning. So cooperation is probably not going to function in the context of helping at the nest in terms of provisioning, but they may still cooperate in other ways, for example, by through group defense, right? So protection of the group against of conspecifics or predators or something like that. So yeah, so they're not cooperative breeders, but they certainly have a social structure from which similar forms of cooperation could emerge. We obviously don't have any examples of a eusocial Egernia. I'm still dreaming of the day that I dig up a big hole in the desert somewhere and we dig up a big queen Egernia, and it's been down there breeding, but obviously that's unlikely. And that was obviously a joke.
Yeah, all right. Thank you very much. Thank you.
No problem.
Lego?
Yes, greetings to everyone. I'm Kulle Wumbanjo. I'm a research assistant at SKRS. I'm studying the external and internal structure of the bush karoo rat. So my question was on the tolerance. So we found that low levels of tolerance from females may lead to infanticide. But I wanted to know if there is any sort of information in terms of what is the role of the males in terms of tolerance? Do they play a role in tolerating the offsprings, and what other roles do they play? But mostly in terms of tolerating, do they have any role to play in tolerating the offsprings?
Yeah. So yeah, so males certainly tolerate the offspring. So typically these social groups will have both males and females, and both are needed to tolerate the offspring. Because it's so easy to bring females into the lab because they're viviparous and they'll give birth, and then you have the females there and you can manipulate things in lots of different ways. They're really easy to get kind of more detailed information on the nature of tolerance and the environmental factors that might kind of promote tolerance. That's not quite as easy with the males because you need to catch the female, you need to catch the male, and you need to obviously ensure that that male is the father of the offspring, and then you can kind of have a look at tolerance. And we've done that once or twice, but it's not as easy as the female, so we haven't done as much of it. The one time where we did do it and had a look at the level of aggression between the male and the offspring, we certainly found higher levels of aggression between the male and the offspring compared to the females, where you get very little aggression. And it was on similar levels to what you see with the siblings. But that was also in an experiment where we manipulated polyandry, so that there may be kind of some explanations for why the males were a little bit more aggressive in that instance, because they, we'd manipulated potentially the relatedness of the litters within which we were kind of placing them in. So yes, so they certainly do tolerate the offspring, but we have far poorer understanding of how that plays out.
All right. Thank you so much.
Thank you. Then we have a question by Suleima.
Hi, can you see me?
Yep.
Great. Thanks so much. I hope you're a night owl, given how late it is where you are. I'm Suleima Tang Martinez. I'm retired from the University of Missouri in St. Louis, in the US. And I have several questions, but I think they're all pretty short, and you might have addressed some of them already. And this, by the way, was an absolutely fascinating talk. I had heard about the lizards, but I did not know too much about them, and so I learned a lot, and it's really a fascinating system. The first question I have is with regards to, you mentioned that divorces sometimes happen, like if one male one member of the pair dies, or I assume like when you were taking females away, that would also be breaking a pair bond. Do you have any idea? I'm assuming that they repair eventually, but do you know how long it takes before they repair? And also, what happens if eventually the other partner comes back?
Yeah. So we obviously remove females usually for a couple of weeks up to a month during the January. And that doesn't seem to disrupt the pair bond. So we still see the stability of the pair bond. One of the reasons for that is that the males are active. The males kind of come out during the mating season, and then they're still around, but they disappear pretty much once the mating season is finished, and you'll still catch them every now and again in the burrow systems with the females, but you won't see too much of them, especially in January where it can get quite hot. And so they're only active for a short part of the day. So that means that the disruption is relatively minimal and we take the females out. Most of the kind of moving around seems to happen in the autumn. So after the birthing period, we've kind of got a pretty good handle on the pairs, and then you turn up after winter and some of the pairs have kind of moved about and changed. So what we think happens, or preliminary data on this, is I think what happens is that female disappears out of a pair bond, a male will retain his burrow system and another female will join him. But typically when a male dies out of the pair bond, the female moves on to another male, if that makes sense.
Yeah.
So we think that males basically control the territories and fight over the territories, and then the females distribute themselves amongst the males. And then if a male dies, then a female goes off and finds another male. Whereas if a female dies, then a male will attract another female, if that makes sense.
Yeah. But in either case, it sounds like it takes quite a while before they sort of go on to repair with somebody else.
It happens usually by the next breeding season.
Wow.
But occasionally, from memory, I think occasionally, I think for the females, there's a lower, they have a reduced chance of pairing than the males do. But that's from memory. So there are usually the next breeding season, but there are some cases where it takes another breeding season before the female, particularly the females, I think, repair.
Yeah, great. Thank you. And then the next question I had, you might already have answered it if the males are not very active when the young are around, but I had been wondering, the presence of the female appears to change the behavior of the young, both in terms of movement and exploration. And I was wondering whether the presence of the father would also have the same effect. But it sounds that that's probably not biologically relevant.
So I mean, I think they probably still play a role, but they're not going to be as active as the mothers in terms of as present. And actually, that's a good question, because I haven't really thought about one of the things that kind of, we tend to, once we've released the offspring, we don't do a lot of work in the kind of autumn period. We tend to go back, and most of our information on parent offspring associations are the following spring. So one interesting thing to do would just to get a feeling on how active or reactive the males and females are in that early period of postnatal association, and see whether there are any differences between males and females in which would suggest some active kind of parental care, I guess. Whereas my assumption at the moment is that they give birth, they're there in their burrow, the offspring are there, and that's the extent of it. But it could be that there's subtle differences where the individuals that have offspring in their burrow system, given that we have huge variation in the extent of tolerance, you might be able to say are individuals that tolerate their offspring more, do we observe them more than those that don't? Because those that don't are, I'm off for the rest of the year, but those that do are kind of, well, I need to be active and present. And that would be some subtle, there would be some nice data that would give you some, potentially insights into the kind of the subtle parental care where it is just tolerance, but maybe mums and dads, are more actively tolerant than non-mums and dads.
Yeah, thanks. And then another question is, I was thinking about some of the birds where you have asynchronous hatching, where you get, sibling rivalry and sometimes even siblicide. Do you see anything like that?
Yeah.
Because in some of the birds, I mean, only one ends up surviving.
Yeah. That's exactly what we see. So it's pretty brutal. The siblings are pretty brutal to one another. I mean, interestingly, they are and they aren't. So we when you kind of look at aggression within the litters, the interesting thing is in 50% of litters on average, the siblings get along fine. There can be 3 or 4 siblings in a litter. It doesn't matter what the birth order is or the birth spread is. They hang around, they bask on one another, they're perfectly harmonious. And then in 50% of litters, and they're just absolute little pains to one another. And not even in a small number of cases, in a lot of cases, if you let that go in the lab where you've restricted dispersal, it is one offspring that will end up killing both of its siblings.
Wow. Wow.
And in the wild, obviously, that's probably triggers dispersal, right? But in the lab, it's as brutal as you get in egrets and raptors
Yeah.
and those kind of things. So certainly siblicide is a key kind of source of mortality in the lab. In the wild, probably dispersal solves that problem, but it only happens in 50% of litters, and we don't understand why it is that some litters are perfectly harmonious and some are full of conflict. Nothing we've kind of looked at has suggested a reason for that.
Yeah, that's fascinating, and the parallels are really interesting. And then my last question, in the polygynous groups, do you see any aggression or conflict among the females that are paired with the one male?
Yes. So females don't like each other. And so I think one of the kind of factors that restricts a male's ability to ultimately acquire lots of female partners is the fact that females don't necessarily get along. So we conducted an experiment ages ago testing this, the kind of environmental potential for polygamy idea, where we manipulated the resources and we provided males in one treatment the opportunity to monopolize a small number of males to monopolize all the resources. And our idea was that would drag all the females together and you could kind of generate polygamy through centering the resources. And then in another treatment, we spread them all out. And we thought we'd get monogamy there because of restrictions in a male's ability to monopolize multiple crevissites. And what we found there was certainly that is what happened, but it wasn't quite as extreme as what we would predict. And I think the reason for that is that even though you kind of bring the resources together, the number of females that control those resources are going to be restricted by how comfortable the females are with one another. And so this aggression between the females probably ultimately constrained the environmental potential for polygamy because they kind of pushed some of the females out to these peripheral environments. And this is a very kind of preliminary result, and something that I need to do a lot more kind of looking into. But I remember years ago when there's kind of two different socially polygynous or ways that males can become socially polygynous. They can either control multiple crevissites, and that's like basically, so you've got females that don't interact with one another, or they can have multiple females in the same crevissite where females do interact with one another. And when I kind of looked at this years ago, and I haven't gone back to it, but I need to, when you have the females, you have the examples of the social groups where you have females that are sharing the same crevissite. When you look at the relatedness between those females, they're all highly related to one another. So one explanation might be that females don't like each other. So if a male is going to become socially polygynous, he needs to basically control two crevicites, which is quite an energetically expensive things to do. Or if he attracts multiple females into the same crevicite, it may be that they are closely related to one another, and that may kind of offset the aggression that the females see towards one another. But that was years ago where I was like, oh, hang on, this could be the explanation for why you get, some form of cooperation between the females in terms of sharing those crevicites with the males, is if they're, potentially closely related.
Yeah. Well, thanks again. Really fascinating. Thank you.
No problem.
Okay, then we have two questions by Adriana, one by herself and the other one that was posted on YouTube.
I think I'll read the one from YouTube just because Melissa's been waiting.
Sure.
Okay, so it says, hi, I'm Melissa, a postdoc studying the evolution of cooperation at the University of St. Andrews. Thank you for the fantastic talk. May have missed this during the presentation, but I was wondering, is there evidence that offspring remain permanently in their natal group? If so, do they forgo reproduction entirely, or do they reproduce? And if they do so, does this occur within the group or elsewhere?
Yeah, so not in the white skink. Offspring tend to remain probably for a maximum of 2 years, and that's before they become reproductively mature. So they mature around 3 years. But in some of these other larger communal Egernia species, so these Cunningham skink and Gidgee skinks. So these guys usually have multiple cohorts of offspring occurring within the social group, up to 5. And there is some evidence to suggest that those individuals then persist and remain in the social group into adulthood, and that they do indeed forego breeding. What happens after that in terms of where they end up, I don't think we have data on for those species. So those species show the most extreme forms of social organization in, I think, probably any lizards. But we don't have the same depth of data that we have for some of these other species. So there's certainly strong hints that they retain offspring into adulthood, and those offspring forego their reproduction, but we don't really know what happens to those offspring afterwards, whether they eventually disperse into a new social group or whether they queue for a breeding opportunity, or what's going on. So yeah, but there's lots of opportunities in those systems. But those systems are quite long-lived and obviously difficult to set up when you have systems that don't mature, species that don't mature for 5 or 6 years. It can be challenging to kind of get at some of these questions.
Thank you. So now I will ask my question. I'm Adriana Maldonado Chaparro from Universidad del Colombia, Universidad del Rosario in Colombia, sorry. And I study social structure mostly. My question is related to recognition. So when you were showing this slide where you had this from social complexity, like in the second step, you had recognition, but I was wondering, what kind of recognition? Are we talking about just offspring pattern, like mom offspring recognition, or any other type of individual recognition? And how is this playing, like what role is it playing in the evolution of social complexity?
Yeah, so I mean, there's a whole arm of a research focus that on the co-evolution, I guess, of social complexity in these groups and communicative complexity. I think, most the evidence, in these systems suggests that recognition probably emerges out of familiarity. So you recognize individuals that you're familiar with. I think that's and some of the things that we were talking about before, especially in the context that you can break that familiarity if you kind of remove offspring away from mum and put them back. You can certainly kind of break that recognition. So I think in these kind of early, the early transitions to these family groups, I think recognition is probably relatively simple and is just based solely on familiarity, which also allows these non-kin groups or non-kin to enter the groups, because you probably have a period of an imprinting period or something similar to an imprinting period that might allow other individuals into the group. But then your prediction would be that as you kind of get these more complex social groups evolving, you might get more complex forms of recognition emerging once you go from just kind of having to recognize your offspring that are always there to some of these larger social groups where you've got like 30 individuals. You need to recognize that perhaps they're going to show more complicated forms of recognition and communication. And that's certainly kind of an area that we're thinking about moving into is this co-evolution of social grouping and recognition mechanisms, kind of focusing also they use olfactory communication and they do use some unique, well, not unique, but specific communicative approaches. So they scat pile. Some of these species will pile up scats at the start of their burrow to kind of signal to other individuals. So there's lots of mechanisms there by which they're kind of sending information to other individuals, but I think that's largely associated with individual recognition, and then recognition is probably the outcome of kind of familiarity. It emerges as a result of kind of being familiar with other individuals. I think there's some evidence that kind of more specific forms of, say, kin recognition kind of occur and kind of underpin some of these patterns, but I'm not sure how confident we are of how sophisticated that is, I guess, particularly in these simple social groups. Or whether you can really separate that from individual recognition.
Yeah, so far we don't know too much about, we might know about kin recognition, but not if they can tell apart one individual from other, or some
Yeah,
other.
no, not really. Yeah.
It's not really. And they would do that basically on olfactory communication, right? Like chemical
Yeah.
signals. Okay.
I would imagine so. They are ridiculously patented. They have kind of quite unique, or not unique, but they have quite variable color patterns on their body, and especially as juveniles, right? And they lose those a little bit as they kind of get older. So yes, I think that olfactory communication is by far the kind of main modality of communication, but I have been thinking recently about the role that visual communication might play in these systems as well.
Cool. Awesome. Thank you.
Then we have a final question by Perfect, his second question, third one.
Hello, I'm back again. I couldn't pick the questions to myself, But I wanted to ask, like, do you have an idea of what role does kin selection play in the stability of these multi-generational groups?
Yeah, so I think, as we were talking a little bit about earlier, I think that probably the formation of these groups, they're obviously kin groups. We get offspring being retained in the social group of their parents in the majority of cases. I think that the emergence of kin structure in these groups is probably a consequences of how these groups form, because females are going to give birth to their own offspring, and any kind of environmental factor that restricts dispersal is going to keep close kin together. Familiarity probably kind of is a main form of kind of recognition, and that is going to be biased towards close kin because those individuals that they're most familiar with are probably close kin. So I think early days, the kind of fact that they're kin groups probably is less a result of kin selection per se and more a result of how these groups form. But I think as these groups become more complex, and the other thing is that because the behaviors, particularly early on, are relatively simple, so parents aren't playing any cost to tolerating the offspring, so maybe they don't really care if offspring are theirs or not. It doesn't really bother them. So that's why other offspring can occasionally kind of end up in these social groups. But I think once those behaviors become more sophisticated and you get the evolution of more complex forms of care, I think the kind of the rules change a little bit, and I think then you might get kin selection playing a role. So the stabilization of these social groups might then be biased towards groups that are full of closely related individuals, because you will get selection against behavioral traits where you're investing a lot in unrelated individuals. So I think early doors, it plays a relatively limited role, perhaps in the emergence, but certainly the stabilization of these social groups, it probably plays a very important role.
Right. Thanks. Yeah.
Okay, thank you very much for everyone for participating in a very interesting discussion, and to Geoff for fascinating talk at a very early time for him. And I think that was a great closure of the second season of Define this spring. I'm stopping with this then the live stream, and I'm still hoping to see everyone who is here and who is watching this later on YouTube, to see you in the fall again, starting the 2nd of